Dear learners, welcome you again. Previously, I have discussed the method of plant propagation, in which I have discussed sexual method of plant propagation, and in asexual propagation methods, I have discussed about the cutting propagation. Now, I start from the layering, which is another type of a asexual propagation method. Layering involves introducing roots and layering involves introducing Now starts with the layering. Layering involves inducing roots on an intact plant. Layering is a method by which adventitious roots are caused to form on stem while it is still attached to the parent plants. Layering causes root to develop on shoots that are still attached to the parent plant. The stem is not cut from the main plant until it has rooted. This method of layering can be broadly divided into two main classes. First one is the ground layering. In this method, the rooting of layers takes place in the ground media or in pots containing rooting media. The next type is air layering. In this method, the roots are introduced to form on the aerial part of the plant where stem is girdled or slit at an upward angle like in case of ficus or in magnolia. Starting from the ground layering. Ground layering is starting from the ground layering, which are further categorized into the simple, compound, trench, mound and tip layering. Simple layering is done by blending a branch to the ground and burying a portion of it while the tip remains uncovered. Treatment with rooting hormone is helpful. And layering is done in early spring while plants are still dormant or in late summer on wood that has not become woody. Other type of layering include compound, trench, mound and tip layering. The simple layering, it consists of a blending down a shoot and burying a part of it in the soil so that the tip is in air. The buried portion is wounded by a single stroke upward on the underside of the shoot to be buried. The layered branch is held firmly in position by pegs and large stone on the surface of the soil above the bending. Roots are formed at the buried portion where the cut has been made. The bent portion is completely covered with soil. The example of this kind of layering technique is used for the rose and jasmine. As we can see, this plant, there is a simple layering, there is a wire peg which is used for the bending the branches. When these branches have a rooting, these are the tip of the plant which are above the ground, and this is the layered branch which is used for the layering technique. The next type is compound or serpentine layering. This is similar to simple layering except that the flexible branches are covered with soil in several places. As we can see in these images, there is a several places this branch is covered with soil. The shoots are alternatively covered and exposed over their entire length. The example of this kind of a method is used in the case of clematis and peperomia. Then trench layering or etiolation method. Etiolation refers to the growing in darkness, and in this method a branch is covered over its entire length to produce from all nodes or buds whether roots are formed. 1-year-old plant is planted slantly at the angle of 30 to 45 degrees centigrade and about 1 meter apart in the ground, as we can see in this particular image. Once these plants are established, they are bent and laid flat over the bottom of the trench of about 5 cm deep, as we can see in this particular figure. When buds start to swell, a layer of 2 to 0.5 soil is placed on the branch. As the shoots grow, more soil is added until they are covered to depth of about 15 to centimeters so that the shoots are etiolated to form roots. These are the different steps of the trench layering as showing in this particular figure. Then the next is mound and tip layering. Mound layering is a form of layering in which the already established parent plant is cut back to the ground and resulting growth from the stud or stool are covered with soil to one half its of height. The soil is applied at the interval as the shoots grow, as in case of apple rootstocks. As we can see, this image showing the moon layering edge when the parent plant is firstly cut back, then it covered with the soil. Then when it is started, the growth of the new plant, like the example of this particular layer is the rootstocks. The next further is the tip layering. The tips of current season's shoots are buried in the soil. The tip of the shoot curved upward to produce a sharp blend in the stem from which roots develop. For tip layering is used for the plant like blackberries and raspberries. And this image showing the tip layering, as we can see the tip of this particular plant is buried in the soil, and when it is started rooting, then there is a development of a new plant. Then the next category of the layering technique is the air layering. Air layering can be used to propagate large overgrown house plants such as rubber plant and bougainvilleas. Woody ornamentals such as azalea, camellia, magnolia, oleander and holly can also be propagated by air layering. For optimum rooting, make air layers in the spring on shoots produced during the previous season or in mid-late summer on shoots from the current season growth. For woody plants, stem of pencil-sized diameter or larger are best. Choose an area just below a node and remove leaves and twig as the Choose an area just below a node and remove leaves and twig on the stem 3 to 4 inches above the below the spine. Air layering differs depending on whether the plant is monocot or dicot. For monocot, make a upward 1 to 1.5 inch cut about 1/3 through the stem. The cut is held open with a toothpick or wooden mark stick. Surround the wound with moist sphagnum mass that has been soaked in water and squeezed to remove excess moisture. Wrap the mass with plastic and hold in place with twist ties or electrician tape. Fasten each and the end of the plastic securely to retain moisture and to prevent water from entering. Aluminum foils can also be used as it does not require twist ties or tape to hold in place. As we can see in this image, the cut has been done on the wood and that it covered with the sphagnum moss and then it is covered with plastic and there it is a holding by the tapes. The process for dicots is similar except a 1-inch ring of bark is removed from the stem. With the sharp knife, make a 2 parallel cuts above an inch apart around the stem and throw the bark and cambium layer. Connect the 2 parallel cuts with one long cut and remove the ring bark, leaving the inner woody tissue exposed. Scrape the newly bared ring to remove the cambial tissues to prevent a bridge of callus tissue from forming. Wrap and cover using the same procedure as that described for monocots. After the rooting medium is filled with roots, sever the stem below the medium and pot the layer. As the root system is small when compared to the shoot system, the rooted layer is planted in pots to produce more roots before planting in fields. This type of method is used for Ficus elastica and Magnolia, and the steps for air layering is showing in this particular image. First, there is a wound into the stem, then apply rooting hormone, then surround this particular plant with sphagnum moss and cover moss with plastic. After rooting, cut off the root ball and plant root ball in a container. The next type of asexual propagation method is division. Division is cutting or breaking up of a crown or clump of suckers into segments. Shrubs may be divided into the same manner, but when the plants are dormant, because shrubs are often woody, they may need to separate it with shovel or hatchet. Each segment must have a bud and some roots. These segments are replanted and grow into new plants. These segments are replanted and grow into new plants identical to the parent. Most perennials should be lifted and divided when they become overgrown and begin to lose vigor. Vigorous growth in most perennial occurs on the outer segment of the clump. Carefully dig the plant, loosening the root and lifting the plants from the soil. Split apart the main clump with two spades or fork, or chop with a shovel or hatchet if the clump is firmly matched. In some cases, outside segment of the plant can be removed and replanted without disturbing the rest of the plant. A good rule of thumb is to divide autumn flowering perennial in spring and summer flowering perennials in autumn. Before planting, trim back the shoot and cut off damaged roots. Shrubs may be divided include red osier dogwood, summer sweet, Clethra dueza, Humnes, Hydrangea, Caria, Asperia, and Snowberry. Herbaceous plants such as chrysanthemum, daylily, peony, iris can also be divided by this method. So this method is very useful for shrubs as well as for the herbaceous plants. The next important method for propagation is grafting. It is an art of inserting a part of one plant into another plant by exposing the actively growing tissues so that they will unite and continue their growth as one plant. Scion is the upper part of the graft and from which stem and branches will grow into plant. As we can see, this is the upper part which is known as the scion, and the base is rootstock. Rootstock is the lower part of the graft and this forms the root system of the grafted plant. Rootstock is also called as stalk or understock. In this image, there is a showing the scion as well as the rootstock. This is the basics of grafting. Then grafting is further categorized into the main four types. The first one is inarching or approach grafting. Next is epicortial or stone grafting. The third one is cleft grafting and the fourth one is the veneer grafting and the last is bridge grafting. Starting from the inarching or approach grafting, in this method root stalks are raised in pots. Then they are brought near to the mother plant. Here scion remains in the mother plant. 1-year-old seedling of pencil thickness are selected. Above ground level at 15 to 20 cm height in rootstock, 5 to 8 cm long slice of bark with wood is removed. This cut should be smooth and it tapers gently towards the tip and bottom. Same type of cut is made on scion and the two cuts are placed face to face and tied firmly with banana fiber and then with the twine over it. After that, union is covered with a mixture of cow dung and mud in equal parts. After 6 to 8 weeks, top of rootstock is removed, above graft union and base of scion below the graft union. First half cut is given and other half cut is given after an interval of 10 days. This image showing the inarching or approach grafting. As we can see, the next method is for the next type of grafting is epicortial or stone grafting. Seeds are raised in a bed and the germinated seedlings of 8 to 15 days old are taken out and grafted indoor by beheading the seedlings about 5 cm above the seed and then inserting the wedge shaped scion in the vertical split at the beheaded stalk. As in this image you can see, this is the 8 15 days old seedlings, and in this old seedlings there is a placing of beheaded new plant. Then after the after that, polythene tapes at 200 gauges thickness are utilized for trying the graft. The grafts are planted in polybags filled. This kind of a uh, grafting is used in case of a mango plant. The next is the cleft grafting. This in this case of the next is the cleft grafting. In this method, the base of the scion is prepared in the form of wedge. The root stalk is split in which the scion is inserted. This method is usually done on thick stalks of 2 to 8 cm in diameter. This is a common method followed in top working of trees. The stalk is given a smooth cut and then it is split at the center and two scions are inserted at the end in such a way that the cambial layer of stalk and scion are in contact, as we can see in this image. After the successful graft union, one of the scion which is well developed is allowed to grow in the garden. Then next type of grafting technique is veneer grafting. This is a modification of side grafting. In this method, a shallow downward and inward cut of 2 to 2.5 to 4 cm long is made in smooth area just above the crown of a stalk plant. And at the base of this cut, a second short inward and crownward cut is made intersecting the first cut so as to remove a piece of wood and bark. The scion is prepared with long cut along one side and very short one at the base of the scion on the opposite side. The scion cut should be the same length and width as those made in the stalk so that the cambial layer can be matched as closely as possible. And this image showing the veneer grafting, which is commonly used in case of the coniferous plants. Then next one and the last is the bridge grafting. A bridge graft is an example of repair graft. It is done to rescue established trees that have been girdled by mechanical damage or animals. Bridge grafting is the best done in early spring when the bark is slipping. Scions are taken from dormant 1-year-old stems. As we can see, this is the prepared scion from the 1-year-old dormant stems, and this is a prepared stalk. Then this is the preparation of a stalk. We can see this is the damaged one and this is the prepared stalk. There is an insertion of scion. Then there is a fitting of the scion, which is known as the bridge grafting technique. Then the next type of axial propagation is budding. Bud grafting is a faster, easier and less messy than other forms of grafting. Cambium layers do not need to be aligned. Bud grafting is done from early July through early August. This method uses newly developed latent bud taken from under a live leaf. Budwood is collected from healthy branches that grew since spring and from young trees because they produce a large amount of new growth. Use buds from the middle section of the branch. The bud is cut from the branch and inserted into T-shaped slice made in the bark of the understalks. Budding should only be done when the bark slips easily away from the tree and it held in the place with special tape or wrap. There is a further various types of budding, which are tea budding, ring budding, chip budding and patch budding. The first is tea budding or shield budding. Tea budding is a common budding type but must be done with dormant scions and active rootstocks. The bark must be slipping. The term tea budding comes from the shape of the cut made in the rootstock, which is the shape like of a tea, as we can see in this particular image. In the rootstock, a transverse or horizontal cut of 1 to 155 cm length is made first. Below or above this cut, a vertical cut of 2.5 to 3 centimeter length is made and connected to the horizontal cut that giving a shape of T. Two flaps of bark should be opened with help of knife. The cuts are given in the stalk at the height of 5 to 25 centimeter above the soil in a smooth bark surface. In the scion, 1.25 centimeter above the bud, a slanting cut is made and 2.5 centimeter long bud is taken in the shape of a shield. Insert the bud by pushing it downward under the two flaps of bark. Then budded portion is covered with plastic tape or adhesive tape. As we can see, this is showing the budded portion covered with the adhesive tapes. Next type of budding is patch budding. It is called so because a patch of scion and root stock is used in this method. In this method, a rectangular patch of bark is completely removed from the stalk plant and is replaced with the patch of bark of the same size containing a bud from the desired mother plant. As one can see, this is the patch opening from the stalk plant and then it is replaced by the scion plant. For this method to be successful, the bark and the stalk and bud stick should be easily slipping. The diameter of the stalk and bud stick should preferably be about the same. Then the chip budding, it is being practiced at the time when bark is not slipping from mother plant. In this method, bark with some wood is removed from but stick as a scion and used for budding. This is the only method of budding that can be done even when the bark is not slipping easily. This method can be used with fairly small material 1 to 2.5 centimeter in diameter. A chip of bark and wood is removed from a smooth place between nodes near the base of the stalk and replaced by another chip of the same size and shape removed from the bud stick and contain a bud of desired cultivar. Grafting and budding can graft scion cultivar onto size controlling rootstock. Many difficult to root plants are graft compatible. Grafting defined by season and type tissue like budding, tea budding, inverted patch, wedge, veneer, whip and tongue. Scion cultivar may be changed on mature trees. Here is the list of grafted and budded plants like apple can be grafted and bud by chip or tea bud cleft, conifers by side veneer, pecans by patch, roses by tea bud or shield budding, and grapes by modified wedges. Then asexual method of propagation also includes some specialized structures like modified stems, bulb, comb, tuber, rhizome, pseudobulb and runner, and as well as modified roots like tuberous roots. This kind of propagation methods have been presented in many plants like in case of tulip, onion, Easter lily. There is a presence of bulb which is natural propagation methods for these plants, and for potato, Jerusalem they have a tuber, iris, lily of valley have a rhizomes, gladiolus, crocus have a corm, strawberry, ajuva have a runner, and dahlia and sylvester have a tuberous root. These are the specialized structures for these plants and they are naturally propagated by these structures. I am going to discuss some of them like bulb and combs. Bulb can be propagated by removing small bulblets or offset that can form at the base of the parent bulb. These small bulbs take 2 to 3 years to mature into plant that flower, as we can see in this image in the case of crocus. This is the corm and it needs 2 to 3 years to form mature flowering plant. Place offsets in rich light soil for their development and this same procedure should be followed for plants which form from combs such as gladiolus. Many lilies can be multiplied by removing scales from the mature bulb. Dust the scale with the fungicide and place base in down in a most growing medium in a warm protected areas. Bulblets will form at the base of the scale. 1 to 4 years, these bulblets will grow and ready to flower. Then next is the tubers and rhizome. These are also specialized structures. Tuberous plants can be dug up and the tubers separated. In separating the tubers, each must have a segment of the crown that contain at least one eye or bud. Like in case of potato and sweet potato, we can see these are the tubers and they can be propagated by the cutting these tubers. Then why for rhizomes? Rhizomes grow and develop buds along their length. Like in the case of turmeric, these are the rhizomes. The rhizomes can be dug and cut into sections that each contain at least one eye or bud. Then they regrow and form a new plants. Last method of micropropagation Micropropagation is the last method of asexual means of reproduction. It has a many synonyms like tissue culture, mass propagation, in vitro culture or cloning. Micropropagation is rapid, continuous and efficient. It is specialized, it requires specialized equipment, facilities and technically trained personal care required. Steps can be taken to obtain and maintain certified pest-free plants. It is cost effective, its large numbers of given clone are produced. Widely used for orchids, ferns and many interior foliage plants and rootstocks. Each plant cell has the potential to grow into a new plant exactly like the parent one. As I have told you, this quality of the plant is called totipotency. In tissue culture, individual or small groups of plant cells are manipulated so they each produce a new plant. A tiny piece of bud, leaf or stem can produce incredible number of new plants in a small space in a short time. The advantages of tissue culture in addition to speed and efficiency of propagation include production of disease-free plants and new plants can be made available to the public more quickly because of tissue culture. Conditions for tissue culture are very exacting. Absolutely sterile condition must be maintained and temperature, light, humidity and atmosphere are strictly controlled with electronic sensor and computerized control. Such costly equipment rules this out for most home gardens. Plant amenable to micropropagation includes flowering pot plants like begonia, African violets, orchids, inferior foliage plants like fern, syngonium, ficus, dieffenbachia, woody ornamentals like red maples, rhododendron, nandina, forest trees like poplar, birch, lobley pine, fruit trees like apple, cherry, pear, vegetable crops like potatoes, celery, tomato, onion, plantation crops like banana, date, plum, coffee. So there's a many amenable plants for micropropagation that make it more convenient for at industry level. Here are the some advantages of asexual propagation. The historical crop which do not produce viable seeds are the horticultural crops which do not produce viable seeds are propagated by vegetative methods. Most of the important food crops are cross-pollinated and are highly heterozygous. When propagated through seeds, the progeny show large variation. So vegetative propagation is remedy for these crops. Asexual propagation methods gives true to type plants. The vegetative way propagated plants bear fruits early. In the case of fruit crops where root stalks are used, the root stalks impart insect or disease resistance to the plant. Vegetative propagation helps to alter the size of the plant, either that is drafting effect. This helps for spraying, intercropping and harvesting of crops easy and economical. By grafting method, different varieties of fruits crops can be grown and harvested. Inferior quality fruit plants can be converted into good quality plants. Through bridge grafting, a repairing of injured plant can be done. Here are some disadvantages of vegetative propagation. Like by vegetative propagation, new variety cannot be developed. It is an expensive method of propagation and required specialized skill. The lifespan of vegetatively propagated plant is short as compared to sexually propagated plant. As all the plants are homozygous, the whole plantation may be get attacked by particular pest or disease. Viral diseases could be transferred through vegetative parts. Thank you all for your participation in this course. Now, this is the last lecture of this course. I wish you great success in your future. Thank you so much.